Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, there's just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jaron Silverberg, head of the Cellus Solution Center at Ajinomoto Genexine.
Thank you, Leah, for the nice introduction. And hi, everyone. In today's webinar, I would like to talk about how you can maximize performance of your biologics or biosimilar cell lines, pipelines, and how you can optimize to get better performance using tools, more advanced tools than the traditional tools being used usually. These things you can do either in your own research centers. Also, we have ways to provide this kind of services in our customer solution center to help the customers. So a few words about Ajinomoto Genexine. We are a joint venture between Ajinomoto, the global food and amino acids manufacturer and developer, and Genexine, which is a Korean biopharma company. And I will quickly show some history about Ajinomoto. Ajinomoto is a Japanese company over 100 years ago established, and it's a world leader in development and manufacturing of amino acids. We already have more than 60 years experience in amino acid manufacturing for pharmaceuticals, and in the past 30 years, there was development of business for cell culture media. We have various media for cells, for stem cells, embryonic stem cells, IP cells, and also mesenchymal stem cells. And more recently, we launched the Cellist products. Cellist are basically media, basal and feed media for production of biologics using CHO cell lines. So Cellist is not really pronounced as cellist, not like a cello player. It's a combination of cell and specialist. And the formulation is based on the long experience and knowledge that Ajinomoto has in the amino acid science. And now I will talk a bit about our other sites around the world, because beside the actual products, we have sites for technical solution and support in Asia. We have our main R&D center in Japan, in Kawasaki, and we have additional customer solution centers in China, in Shanghai, and in India, and the main one in Korea. The one I'm heading here is in Korea. We support not only Asia, but also European and US customers of our cell culture media. So, quickly overview of my talk. I want to briefly explain a bit about the biologic biosimilar drug market and to show a general workflow for evaluation and development of cell culture media that we usually employ with our customers. And I especially want to give some tips and pointers about how to ensure cell culture performance and high productivity for your cell line, especially in the terms of adaptation and how to switch from one medium to another. Now, I will mainly focus later about the optimization and what kind of methods you can use to optimize both the importance of feed manner optimization and using of DOE, design of experiment approaches for media formulation, and later on about more advanced ways, multiomics and digital twins or biosimulations that are kind of more advanced ways to find metabolic bottlenecks in the process and to find which media components are more critical than others. And finally, I will present some new supplements that we are developing now for enhancing productivity and for making a more ease of use when introducing feed to your process. Okay, so first of all, about the biologic and biosimilar market, as you know, it's a really huge market, and just last year the revenues was over $300 billion US, and this market is projected to double by 2030. And you can see that the 10 top drugs or biotherapeutics are mainly against various cancers and other autoimmune diseases. For example, Keytruda is a biologic by Merck, is projected to become #1 seller, best-selling in the next few years. It's an antibody that targets various types of cancer. And beside those biologics, there are a large amount of biosimilars that are competing with the biologics. So slowly, biologics drugs are expiring, which means dozens of biosimilars are competing with each of the biologics that are expiring, and then it makes the market basically grow quite quickly. So basically, the biologic drugs manufacturing workflow looks like this. You are probably familiar with this kind of chart. The cell culture media part looks a bit small, just the initial stages. But these stages are the most important because in these stages, the cell grows, all the biomass grows, and the drug is being produced. And that's why this is very crucial stages, and the influence is very strong of the cell culture media on the overall productivity, as well as downstream purification steps and even on protein quality. So when we work with our customers for helping them choose the right media or develop media together with the customers, we follow a general workflow that looks like this. The evaluation of the different types of media has to start with some adaptation. So it's really important that cells are adapted to a new media before there is some kind of evaluation. And I will explain more about this later. Then there is batch and fed batch. Usually fed batch is directly examined. You don't have to actually do a batch culture first, but it's easier to select basal media and then select the feed which combines with it well. Then it follows to basic optimization of various things, for example, the feeding manner, different supplement and process parameters like pH and temperature shift, things that can increase the titer, the productivity. And finally, something I will talk about more in this webinar is the advanced optimization, how to target specific components and how to set the right combination and composition of amino acids and other supplements. And also there's targeting of protein quality considerations such as glycosylation and charge variants. So I just want to briefly present a few pointers that will help you to ensure you get the maximum performance from your CHO cell line. And this looks quite obvious. Most of these pointers are things that you already know, but some of them are quite important to take into consideration. That's why I want to mention them. The first two steps of recovery and then sequential adaptations are very crucial in order to evaluate new media. You need to let the cells get used to some new media before they can get the best potential of that media. And I will show some example later. Other issues which are important are quite cell line dependent, so it depends which cell line you use, but additional growth factors will really help the initial growth and the adaptation to the new media, as well as glutamine. And consideration should be taken care also about glucose, and not too much, not too little, and effects on osmolality, and also adequate oxygen transfer in order to ensure the level of oxygens are good enough for the culture. Okay, for media switch and cell adaptation, it is important to remember that we want to ensure correct adaptation to the new target medium. Why do we want to perform a media switch? So the main reason is that the original cell line medium that was used during the cell line development might not be the optimal medium for that cell line in terms of the growth and the productivity. Other medium might actually give better results and better titer, better productivity. There's also other consideration, of course, of supply and tech support of the company and other logistic consideration, and of course, cost consideration that makes a biopharma company just switch to another medium. The consideration for switching is quite important. Important to remember that the original medium is the medium which in the cell was developed cell line was developed in, the cells are used to those media, and we need to ensure a gradual adaptation into the new medium that we want to adapt the cell to. So here I would like to throw a question to you, the audience, and I would like to ask you, if you are performing some kind of media screening on your cell line, when you are performing this screening media for your cell line, what is the main difficulty that you encounter, the main issue that you care about or that troubles you, or your main priority for the switching? So is it A, you encounter issues of cell adaptation and growth in various media you test, or B, achieving the desired target protein titer, the desired productivity that you set as a goal for this pipeline, or C, issues of protein quality variations? Are you worried about variations in glycosylation or charge variants? D, any other reason that you might have when considering new media? So I will let you answer if you can. Okay, thank you for your feedback. Next, I would like to quickly mention about the adaptation. So probably you are quite familiar with this kind of general approach for increasing concentration of target medium compared to original medium in order to ensure a gradual adaptation of the cell line. So some cell lines can be adapted quickly. Some cell line required only maybe 2 switches from 0% to 50% to 100% of the target medium. But some cell line require more gradual adaptation if they are more sensitive. Just for example, I will show you this kind of graph that shows how sequential adaptation affects the culture performance. The variable cell density, the VCD, was measured during a main culture, and the green line shows you cells that did not go through any adaptation stages, and it goes in the target media. The red line is actually the same cell line going in the original media. You see the VCD, the biomass doubles and even more when there is no when there is original media compared to no adaptation. But if you look at the blue line that shows a 2-step adaptation that was performed prior to the main culture, you can see that only 2 steps, 2 passages of adaptation prior to the beginning of the main culture already makes the VCD profile matching, almost matching that of the original media. So they were enough to really ensure that the cell were growing well in the target media. So both the green and the blue and the orange lines are actually in 100% target media, but they vary in the passages of adaptation they went through before day 0. So that's basically to show you the importance of the adaptation prior to selection of the basal media you would like to use for your process. So after selecting the basal media, you also should choose the feed, and it's important to optimize the feeding manner. And there is a basic way to do it by varying the volume or the percentage of feed that is added. Usually anywhere between 2 to 10 is suggested by various manufacturers of feed media. We usually suggest between 2 and 6% for our feed medium. It's important also to remember that some cells will grow much better in a low percent, only 2%, and some will be going better with 6% feeding. In this example, 2, 4, and 6% were the condition that was checked, and also starting date varied between day 3 and day 4. And in the results, in the lower right columns, you can see that there's a big difference between 2 and 4%. So 4% definitely shows much better productivity, tighter the protein production. Red and green means starting day 3 or 4 doesn't make such a big difference, and also increasing to 6% didn't improve the titer. So we can assume the actual optimal feeding manner is between 4 and 6%. Now, there is also the issue of varying the amount of feeding every time point, what's called dynamic feeding or variable feeding, but on that I will touch later. So for the next step, the question is how we can increase the titer by optimizing the actual media composition. So of course it's impossible to actually screen all more than 50 or sometimes 60, 70 different components, different raw materials that are composing the cell culture media. It takes too much time and too many resources to do that. So it's not feasible. So what's important to do is to select the key candidates and those that believe to be bottlenecks in the process or limiting factors, and they will greatly affect the culture performance. That could be any component, amino acids, vitamins, metals, or others. DOE, design of experiment, and statistical analysis using computer modeling and multi-omics approaches usually help identify those specific metabolic pathways and components that allow to increase the culture performance. So now I have another question for you, the audience, and I will really appreciate your feedback because I'm interested to know if you are testing media and trying to develop media using this kind of procedures or tools. So my second question for you is, what type of procedures or tools do you usually apply for media selection and optimization? Is it A, just screening basal and feed media using shake flasks or other equipment, or B, screening basal and feeding media using bioreactors like the Amber microbioreactor system, for example, or benchtop bioreactor. C, screening for media and also applying design of experiment and statistical analysis such as factorial designs or other. And D, using advanced omics, metabolic analysis, or other type of biosimulation techniques in silico. So thank you very much for the feedback for that. And next, I want to just quickly show an example of a DOE, design of experiment, that can be easily employed. We can do this kind of screening using what's called a Plackett-Bowman DOE design that allows screening of large number of factors. In this example, all the amino acids were included, each of them with a higher and lower dose compared to the original media. Plus means a higher dose and minus is a lower dose compared to original media. So all 22 amino acids were actually included in this test. It includes 24 designs, so 24 vessels, 24 bioreactors needs to be used. And one problem with this design, though, it cannot take into account any synergetic effect or combinatorial effect between factors. It can only measure individual contribution of those amino acids. But it may be useful to do this because it can target a few amino acids or other components that specifically for your cell line have an influence. And you can easily adjust those amino acids and then do validation experiments. But for more precise analysis, you want to minimize the number of factors, and you can perform this kind of factorial design that is shown on the left. This is a 4-factor, 2-level full factorial design that examines the effect of 4 amino acids. So I'm sorry, this part is a bit confidential, so we just wrote amino acid 1, 2, 3, and 4. And I will show you quickly the workflow that we employ in our lab. We start on the top right equipment called the Junior. This is a robotic automated powder dispenser, and it allows to create any kind of powder combination we want to make. And then we formulate all the different designs and use the Amber, the automatic microbioreactor system, to perform a 2-week culture, fed batch culture. Then all the data is being analyzed, all spent media analysis, including all amino acids and also VCD, viability, and IgG production. And all this data is inputted back to statistical software, for example, the JUMP software, in order to analyze correlations between the factors. So initially, you can directly see some effects that a few designs like number 4 and number 8 in this graph shows higher blue columns, which mean titer. So increase of up to almost 20% in titer was found for a few designs of amino acids. So that's already some improvement that you can get right away. But more important is to understand the synergetic effect between amino acids and the power of statistical analysis using the JUMP software allows you to use this kind of data to find synergetic effect. For example, as you can see on the right hand, two amino acids, amino 1 and amino 2, and the bottom right graph shows you the IgG titer, and you can see that they have a strong synergetic effect for increasing the productivity, the titer for this cell line. Now, it's important to remember these effects are really depending on the media, because other components in the media could be affecting the productivity too. And it also depends on the cell line. Every cell line, every cell clone behaves a bit differently. So it has to be optimized per cell clone. So this is basic DOE, which been used for years. More recent developments were done using the omics that I mentioned before, multi-omics and advanced computer modeling, which is used for both media and process development. So the traditional optimization with DOE is mainly taking the wet experimental data and performing a lot of statistical analysis and correlation analysis on it. But all the new concept of the omics is to integrate all the statistical data analysis together with mechanistic models, metabolomics, genomics, a genome scale model of the cell. And the advantages of this kind of analysis is because those models give a better representation of the cell. All the metabolic pathways and biochemical reactions that are known from previous knowledge from the literature are incorporated in this model. We can use it to identify rate limiting steps, the bottlenecks inside the cellular network, and we can find specific components that can bypass those limitations. Beside effect on performance and productivity, you can also use this kind of modeling to control or predict protein quality characteristics, such as the glycosylation profile or charge variance. Just to show you how powerful this kind of analysis is, this is a kind of visualization of how it works. First, you can see different amino acid consumption rates, which are analyzed on the top left window. Those are calculated from a wet experiment that was done on a fed batch culture. After you analyze the spent media, you can feed all these amino acid concentrations and consumption rates into the model, and you really can visualize and get information about the flux rates or which metabolic pathways are highlighted and have more importance in the overall process for this cell line. And more than that, you can actually pinpoint, as you can see at the bottom left side, you can pinpoint specific biochemical reactions, and the model employs more than 7,000 of those individual biochemical reactions, and you can measure the effect of components on the rates of reactions. So for this kind of analysis, we are having a long-term collaboration with the group which is led by Professor Dong-Yap Lee from SKK University in Korea, in South Korea. They are leaders in the genome scale modeling specifically for CHO cells, for CHO cell line. Their model basically incorporates hundreds of metabolic pathways in over 7,000 of biochemical reactions, as I mentioned. And this whole model is based on actual literature publications about experiments and about general knowledge, global knowledge. And a few communities around the world together contributed to make this powerful model, which is now existing. So this is a bit complicated slide, but I will just quickly go over the main points. At the top left side, media development, you can see some initial wet experiment that was done. Two cell lines, two CHOK1 lines, 1 and 2, were cultivated in two different types of media, A and B, which gives 4 different conditions. And the target, the point of this analysis was to find which components are more crucial in the media and how we can improve the productivity. So after collecting the information and getting growth profiles for VCD, for example, variable cell density and specific growth rate, and also protein production, what you can see on the bottom left side, the cell culture profiling and the data processing, this is part of the media characterization part. This is followed by a statistical model. The data-driven statistical analysis includes MVDA, so multivariate data analysis, and other ways to correlate between different components in the media and metabolic pathways, and to narrow down the important factors you want to target here. And the final stage is the mechanistic model. So on the right, this is a model-guided analysis and media component targeting. And in that part is based on the actual genome scale model and the metabolic flux rate analysis. So I will quickly explain the statistical and the mechanistic analysis in the next 2 slides. The first part, the data-driven statistical analysis, is basically using the VIP scores, the variable importance score, which allows to kind of rank all of the components that participate in all of the pathways in the cells and find the most important one, the crucial ones that contribute the most to the model. So the VIP score is kind of a contribution measurement of the component to the model. A similar thing is done for the pathways itself to find which are high impact pathways. So I'm not going into the details here so much now, but just want to quickly give you the overview of this. After narrowing down the important pathways and factors that may contribute more greatly to the model, what is being done is the mechanistic analysis. So the genome scale metabolic flux distribution shows here various pathways, but in this example, for example, we saw that there is a limited pool of FAD, FADH, as you can see in the bottom square, the blue square. Low amount of regeneration of the FAD, FADH, specifically in medium A, suggested that we should focus on the electron transport chain, which is the left window. And the left blue square shows the electron transport chain. And this analysis identified the key component, the coenzyme Q10, and suggested this component for enhancing overall cell growth. So following this outcome from the modeling, a wet experiment followed. And as predicted from the flux analysis, it was shown that especially for medium A, which is in this figure, number A and C, the left 2 charts using medium A shows much increase in cell growth, more than 30% in one case. That shows a increase in the VCD for that cell line. But actually for 2 cell lines, but for 1 cell line in particular. So I'm sorry, I'm going very quickly on that, but I would like to just mention that you can see this publication soon. It will be published in Metabolic Engineering. Now, beside the actual media development that can be used using the omics analysis, we are also developing more specific customer services And for that, we are using the services of a company called In Silico Biotech. We recently formed an alliance with In Silico Biotechnology for the digital twin bioprocess simulation. This joint service gives customers a rapid tailor media and feeding strategy optimization. And the main idea is that the customer can give us the cell line or perform the experiment by themselves and give us the data or the spent media for our analysis. Only one set of experiment is needed. This data can be given to In Silico, and they will produce a digital twin bioprocess model and use it to optimize the media formulation as well as the feeding protocol, the feeding manner. And that could be done in a matter of weeks. So you can optimize your media and your feeding in a matter of a month or less than 2 months in total. The dynamic feeding manner that employed really can increase the titer, the productivity. And just for example, we took a cell line which feeding manner was already optimized between 2 and 6% volume feeding, and we employed the digital twin analysis on that cell line for changing dynamic feeding manner. And the increase was over 25% of productivity just by changing the feeding manner itself, even without touching the formulation. If you are reformulating the media, you will get, of course, even higher titer improvement. And last slide, or almost the last slide, I just want to quickly mention about considerations that are important when you are modifying media components. I mentioned before, osmolality and pH, solubility are all really important. Stability of components that you change the concentration is also important, and the cost. Of course, the availability of raw materials is important, especially these days during the COVID pandemic, and now with the war in Ukraine, you have a lot of supply issues. So you need to consider these issues and look for supplier that can give you a shorter lead time. For stability issues, oxidation of components such as cysteine, that's an important issue that we are also targeting by developing new supplements. And the supplements that we are developed recently is the cyst supplement, which includes a component to ensure the cysteine is not being oxidized and give more stable supply of cysteine to the cells. Two products that we are releasing soon is cysteine 1, cysteine 2. Cysteine 1 is basically a feed supplement that can be used with any type of feed, with our feed or with other manufacturers' feed. And cyst 2 is more specifically for a two-agent feed, like Cellbus 7, for example. The main 2 points of this feed supplement is increase of titer. So we saw on one of our cell line up to 27% increase just by adding that supplement. And as you see that the graph at the bottom right side shows you the availability of cysteine or the concentration of cysteine. So lower level of cysteines are better because cysteine being oxidized to cysteine means there's less availability of cysteine to the CHO cells. The red and blue lines there shows you the lower level of cysteine that happen because addition of this supplement to the feed medium. And finally, I just want to quickly discuss about our customer service center, which is located in Korea with the new branches opening now in Shanghai in India. We give all those services that I discussed before to customers, either remote help or cell adaptation and cell banking and screening services also. More than that, we are employing the new tools for developing media using in silico and multiomics and computer simulations in order to improve media per customer. Also, we provide protein quality services and scale-up support to our customers. And finally, I just want to thank everyone who took part in this webinar and the projects that I mentioned, CSC members and AGX members who helped to make this webinar, the India Customer Service Center, the main R&D center in Japan, and and the people from Professor Dong Yap Lee from SKKU University, and people from In Silico Biotechnology. So thank you everyone for listening to my webinar. And if you have any questions, please let me know. And you can always send me an email at the email address below. Thank you very much.
Great. Thanks, Yaron. So we do have
Thank
some
you.
questions for the audience, and as a reminder, if you'd like to enter your questions for Yaron, we will try to answer them, and if we don't get to them, we will be passing them along for him to follow up with you directly. So the first question is, can the culture media be changed into another manufacturer's media at any stage of the process?
That's a really good question. So the medium can be changed at really any stage, and even after going to clinical stages, um, you, the medium can be then changed. Some regulation issues need to be employed, and maybe a process change has to be applied for regulatory reasons. But there's never any problem, especially when you're changing from one chemically defined media to another. And we always provide full support for media switch for our customers.
Okay. And what advantages does your media have compared to other manufacturers?
Mm. So our media has a few advantages. Quality wise, our media has a really long history of amino acids optimization over many years. Beside quality, we are very flexible for customer requirements, and the lead times are really short. And we really use this kind of advanced tools that I mentioned here, like the multiomics and metabolic analysis, to specifically tailor media to our customers. Also, our collaboration with the In Silico Biotechnology allows this optimization to be really quick, very fast time. In less than 2 months, you can really customize and optimize media. I think that's the main advantages. Mhm.
And what is the highest productivity achieved using your media, and can we receive samples for testing?
Yeah, productivity, it really depends on the cell line, of course. But we have currently we employ some CHOK1 lines who are providing more than 8 gram per liter, 8 gram per liter of antibody titer. And we are also able to improve 2 or 3-fold on a cell line that started much lower. So we can really basically optimize the titer efficiently. And you mentioned about samples for testing. So of course we can give free of charge samples for testing of both basal media and feed media for our customers.
And speaking of cell lines, is the media available for other cell lines?
Yes. So we are actually developing now cellulose media for HEC cells, HEC293 cells, for production, for example, of a coronavirus vaccine and other factors. So we will soon have media for HEC cells, and already now we have also media for stem cells, MSC cells, and IP and embryonic stem cells as well. Yeah.
Okay. Is it recommended to add supplement to encourage cell growth, such as L-glutamine and growth factors?
Mhm. Yeah, that's a really important point, and I think I mentioned it earlier in the presentation. It depends on the cell line and the cell clone, but generally, especially if you try a new medium on your cell line, it's really recommended to add glutamine to increase the initial growth, because glutamine will have very strong effect, and when the media is changed, it's a strong shock on the cells. So it's better to help the cell adjust to the new medium. Other growth factors are usually helpful to add, but it depends on costs. So as you know, growth factors are a bit more expensive. For glutamine, Ajinomoto also produces dipeptide with high stability, the alanine glutamine dipeptides that we recommend to use to replace the glutamine, which is less stable.
Okay, well, we still have a few more questions, but I think we just have time for one more question. If we didn't get to your
Hmm.
question, we will pass it on to Yaron. He can follow up with you directly, but also you also can continue to type in questions, and we'll pass those on as well. But the last question for the webcast is, can your feed media and cyst supplements be combined with any basal and feed media?
Hmm. Yeah, that's a good question, and yeah, we can combine both a tea supplement with any kind of manufacturer's feed media, and also our feed media can be also combined with other basal media, but we strongly recommend using both our basal and feed because the feed was developed specifically for our basal and probably will ensure the best, highest results if you use both basal and feed.
Okay, well, thank
Yeah.
you, Yaron, and thank you to our audience for joining us.
Thank
Our
you very much, Leah. Thank you.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you. Goodbye. Goodbye, everyone.